What is microbial genetics? It is a subject within a microbiology and genetics engineering, and also microbial genetics studies microorganisms for the different purposes. Microbial genetics has revealed that genes consist of DNA, an observation that laid the foundations for molecular biology. Subsequent investigations of bacteria revealed the presence of restrictions enzymes that cleave DNA at specific sites, giving rise to DNA restrictions fragments. Amplification of specific regions of DNA also can be achieved with bacterial enzymes using polymerase chain reactions or PCR or other enzymes-based methods of nucleic acid amplification. Genome sequence data have been indicated that most prokaryotic genomes consist of a single cellular DNA molecule containing from 580 kpb to more than 4,600 kpb of DNA. Bacteria may also contain extrachromosomal genetic elements such as plasmids or the bacteriophages. Many bacteria contain additional genes on plasmids that range in size from several to 100 kbp. Plasmids are small genetic elements that replicate independently of bacterial chromosome. Most plasmids are cellular, double-stranded DNA molecules varying from 1,500 to 400,000 base pair. DNA circle, chromosomes, and plasmid, which contain genetic information necessary for their own replication, are called replicons. Bacteria usually have only one copy of their chromosomes. They are therefore haploid. Because bacteria have only one chromosomes, alteration of a gene mutation will have a more obvious effect on the cell. Transposons are genetic elements that contain several kbp of DNA, including the information necessary for their migrations from one genetic locus to another. The involvement of relatively short transposons, 700 to 2000 bp long, known as intersections elements, produce the majority of intersections mutations. This intersections element, also known as intersection sequence elements, carries only the genes for enzymes needed to promote their own transpositions. Almost all bacteria carried IS elements, with each species harboring its own characteristic ones. Viruses are capable to survival but not growth in the absence of a cell host. Replication of the viral genome depends upon the metabolic energy and the macromolecular synthetic machinery of the host. Distinctions are frequently made between viruses associated with eukaryotes and viruses associated with prokaryotes, the latter being termed bacteriophage. Bacteriophages are bacterial viruses. These extrachromosomal genetic elements can survive outside of a host cell because a protein coat protects the nucleic acid genome. Many pages contain double-stranded DNA, others contain single-stranded RNA, and some contain single-stranded DNA. Bacteriophages infect bacterial cells and either replicate to the large numbers and cause the cell to the lyse or lytic infections, or in some cases integrate in the host genome without killing the host. The lysogenic state, such as E. coli bacteriophage lambda. Bacteriophages lambda remains lysogenic as long as a repressor's protein is synthesized and prevents the phage from becoming unintegrated and replicating independently of the host chromosomes. These reactions can be triggered if the host cell's DNA is damaged by radiations or another means of if the cells can no longer make the repressor's proteins, a signal that the host cell is unhealthy and is no longer a good place for freeloading. The change of genetic material between bacterial cells may occur by one of the three mechanisms: conjugation, transformations, transductions. Conjugation is the mating or the quasi-sexual exchange of genetic information from one bacterium to another bacterium. Transformations, which result is acquisitions of new genetic markers by the incorporations of exogenous or the foreign DNA. Transductions is the transfer of genetic information from one bacterium to another by a bacterium phage. Once inside the cell, a transposon can jump between different DNA molecules, example the plasmid to plasmid or plasmid to chromosomes. Transposition, moving part of bacterial DNA from chromosomes to plasmid, vice versa. Only plasmid can be shared with another bacteria like 1A gene.